Okay, so when it comes to ATPase drug therapy, well, the large protein here it is a large protein, and it has 120 and 35 kilodalton subunits. The alpha subunit has 10 transmembrane helices with large cytoplasmic domains. Obane and digitalis bind to the alpha subunits, preventing any sort of conformational change. We're talking about a conformational change in that E1 to E2 transition. So those are two drugs, which I've got down here. These are two drugs, which, heck, what does this look like? That looks a lot like cholesterol. So these are almost cholesterol derivatives that they're going to be able to bind and prevent this transition from taking place. So what that's going to do is lead to an increase in intercellular sodium concentration. So that sodium is, if we're preventing that conformational change, we're not going to see any sodium that is in the cell. Any sodium that is in the cell be pumped out. Okay. Thus stimulating the sodium-calcium antiport, increasing the concentration of calcium in the sarcoplasmic reticulum. Therefore, calcium is released during cardiac muscle contraction, which intensifies that contraction. Another drug, palitoxin, binds to the transporter, keeping it open. So you have that continuous movement of those ions. So next up is an ion gradient driven active transport. So systems like that sodium potassium ATPase generate electrochemical gradients across membranes that can be used to drive endogynic physiological processes. The intestinal epithelial cells take up glucose by a sodium ion dependent symport. The uptake of glucose is a secondary active transport because the sodium ion gradient is maintained by this sodium potassium ATPase. The sodium ion glucose transport system concentrates glucose inside the cell. So we're going to get into this a little bit more, but basically what this shows are secondary active transport. We've got our sodium potassium pump right here. That is going to help us maintain the proper levels of potassium and sodium inside the cell. What we also have is in the intestinal lumen, what we want to do ultimately is we want to get glucose from the intestinal lumen into the blood. So how do we do that? Well, we have this symporter that takes two sodium ions and a glucose molecule and transports them into the epithelial cell. Once in the epithelial cell, you've got to get the sodium out of the cell, so you're going to use our our sodium potassium pump that is going to bring 2 potassium ions into the cell and take 3 sodium ions outside of the cell. Okay, the next one is our glucose uniporter, or GLUC2 protein, that is going to move this glucose from the epithelial cell into the blood so that it can be pumped throughout the body so that it can ultimately be metabolized. But that about sums it up. I hope this is helpful. I'll see you in our next and our last video. Alright, have a good one.